think it through, decide on the whatever, and then leave it aside. You can't just not think about it.
It's the lower though.
We essentially have 3 more regular lecture exams, but the last exam has an additional 50 questions that are comprehensive from the previous 4 years.
So 3 more exams
The easiest
total?
way to, 3 more total, yes. So the last one is during finals week. And it's like 125 questions worth 200 points.
So we have 400 points.
Yeah.
For the lot, or for the lot.
We have points, yeah. 250 for the last one. And then 100 points total per student. All right, so if you'll open your lab manual to page 10, where are you here? 104, 105. So even though we're only being quizzed on the spinal cord on Monday, this material obviously will be on the lab exam and lecture part, all right? But the spinal cord itself will be the quiz topic for Monday. So on the top of the diagram on page 104, you can see the 3 different types of neurons by shape that we covered briefly during lecture. And you can see, actually, I'm going to move this one over. The glare on this one back here is difficult. And everything that's on the smaller side is on the larger chart here. Alright, so remember I said in your lab manual you would have the term unipolar. Alright, this is actually a true unipolar neuron right here. But you don't see that diagram in your lab manual. So multipolar is shown as this type right here. And that's our typical motor neuron that we find forming the motor nerve fibers, axons in our peripheral nerves. And what structure in the spinal cord would be made of the cell bodies of the motor neuron? What structure that that's on your diagram? There's 2 areas that have, well, one area that would have multipolar like this, and the other area is similar to it. 2 areas that have motor cell bodies. Ventral gray horn, all right, and then the autonomic motor cell bodies on the lateral gray horn, okay. And we see this just in the thoracic level, thoracic and lumbar level. There are autonomic cell bodies in the sacral, but it's not a prominent lateral gray horn. All right, then we have the bipolar neurons. Those are sensory, but we find them really in just 2 classic areas. So when we talk about the eye and the ear on our last day of lecture, for this unit, the photoreceptors are the rods and the cones. And so those are just receptors. They don't create an action potential. They trigger the neuron's action potential. And so the cell that they synapse with, it'd be like if I were to touch the table, I'm triggering the Meissner's corpuscles, and those will synapse with the sensory neurons whose cell bodies form our dorsal root ganglion. So there's a sequence. So in the eyes, the rods and cones, when they are triggered, will activate bipolar neurons in the retina itself, and then those will synapse with other neurons that are forming our optic nerve. So the retina is one location where we find bipolar neurons. And then in the olfactory epithelium, the roof of the nasal cavity, just under the cribriform plate, we have cells that have chemoreceptors for modifying and processing those chemicals into smell. And they also synapse with bipolar neurons that pass through the cribriform plate and then form the olfactory nerve, other neurons, synapse with neurons that form the olfactory nerve. All right, now the bottom lower right corner here. You can see the progression from bipolar to the formation of the pseudounipolar that I diagrammed during lecture. And these are the most common of our sensory neurons. And their cell bodies are going to form what structure on the diagram that you have? that Matt passed out to you. What structure is formed as a collection of sensory cell bodies? The ganglion, so the dorsal root ganglion, that's what it's formed of. There's a whole bunch of cell bodies that look like that. And then we also have cell bodies in the posterior great horn, but they may not necessarily be bipolar. Okay, certainly your dorsal root ganglion cells are going to be the pseudo-unipolar neuron. So on your lab manual, then the letter H would be bipolar. Letter I, if you would put pseudo-unipolar instead of unipolar. And then J would be multipolar.
Can you repeat those?
So H would be bipolar. I would be pseudo-unipolar.
And then that's where we cross it.
Right, instead of the, so unipolar you can leave, it's just I want you to know that that looks like this. And the diagram would be the pseudo-unipolar in your lab manual. So just add that word. And then J would be multipolar.
Thank you.
Okay? Now we've talked about the peripheral nerves and cranial nerves, we talked about the facial nerve and trigeminal and femoral and tibial and so on, but not as a structure. And so we'll be doing that in lecture on Monday, but since it's on this page, I'll just go over it now. We would have covered this with muscle if we'd done that last lecture, so the epimysium, myomysium, endomysium rather, and perimysium we would have covered. And it would just been a repeat, but here is showing one peripheral nerve that's cut in a cross section. You're seeing just part of it. So let's say this is the sciatic nerve. When you touch the sciatic nerve, just under the piriformis, you would be touching this outer covering. It's collagen fibers and fibroblasts. So it's just a regular, dense, regular to irregular connective tissue. And this is called the epineurium. So that would be letter A on the diagram in the lower left of your lab manual. And it's formed of thousands of individual axons and peripheral processes of sensory neurons. That's what each of these structures are. Like little donuts, some of them are myelinated, some are not. And so forming a fascicle or a bundle of 20 to hundreds of these is another connective tissue layer known as the perineurium. And then around each individual neuron is a soft, wispy, almost like a spider web of loose, irregular connective tissue called the endoneurium. All right. So the perineurium is going, the epineurium is A, the perineurium is B, and C would be the endoneurium. But it's so difficult in tiny space there that I wouldn't tag it on that. The endoneurium you can see on a couple of our nerve models. So this is a model of a neuron. They couldn't fit it all on one board, so they took the cell body and put it separately. So this axon is continuous here. And then it's covered by a layer of myelin sheath. And then this cement-looking material is supposed to represent the fibers of loose, irregular connective tissue. So it's outside the neuron cell. All right, and I'll go over those layers in a moment because the students a lot of times think this is part of the nerve cell, and it's not. It's just the loose, irregular connective tissue supporting little capillaries and structures among individual nerve cells. All right, so that endoneurium would be letter C. And then D is shown enlarged as this myelin sheath structure of the peripheral nerve. Now, if this is a Schwann cell, that tells you this is a peripheral nerve. All right, the oligodendrocytes do the same function, but inside the spinal cord or inside the brain. So I've taken the diagram on the lower left of page 104 and redrew it up on the board. So I'm going to move over there because I want you to add another term. So this is a longitudinal section, back to that word, of an axon similar to what you're seeing right here. Okay. So here's the axon that's been sectioned here. And like any cell, it has a plasma membrane. So it's just a continuation of the cell body as one long process. So the plasma membrane covering that axon is known as the axolemma. If you would add that word. That's where we find the voltage-gated channels that I talked about that are propagating the electrical current down the axon to its terminals. Then we take a Schwann cell. So one of these structures right here between the two nodes is a Schwann cell. And out on the tables is a model that's probably a little easier to understand. Though it looks kind of weird at first because it's painted into the background. But here's the cell body, and here's one Schwann cell, and here's another. All right, another name for a Schwann cell is a neurolimocyte, but I like Schwann cell. So here's the axolemma. The surface of the blue would be the axolemma. And this is the Schwann cell, and here's another Schwann cell. So we didn't have time to talk about it much in lecture this morning, but imagine, remember the entire Schwann cell is going to wrap around one axon, not multiple axons like the oligodendrocyte does. It doesn't have octopus-like processes. So imagine taking a sleeping bag, a down sleeping bag or whatever, and you're going to roll it up. You're not going to put it in a stuff sack. You're going to roll it up nice and tight. And so to wrap it around, you put a big fat broomstick in the at one end. And that is representing the axon. And then you take your sleeping bag and you start rolling it around that broomstick, squeezing the air out as you go along. And it starts to poof up the end of the sleeping bag. All right, so the sleeping bag is your Schwann cell. The axon is the broomstick. And the layer that you're laying really, really, really tight is the layer of plasma membrane forming the myelin sheath. The poofy area at the end is going to wrap around, and that's the neurolemma. All right, so let's put it back on go back to the board. So this is the Schwann cell wrapped around the axon. The axon's not part of the Schwann cell, but the Schwann cell is wrapped around it. The neurolemma is the outer plasma membrane of the Schwann cell plus that leftover cytoplasm. And then the myelin sheath are the very tightly wrapped layers. So it takes both of those to form the Schwann cell. The neurolemma plus the myelin sheath equals Schwann cell. All right? Now, on the lab quiz or a lab exam, then I have to be specific about what I'm asking. Because if I just put a sticker here, you wouldn't know if I wanted Schwann cell or neurolemma. All right? So I'll have to say name this cell, or name this membrane, or name this layer. Then it's a little bit more specific and you have a better idea of what I'm looking for. The gap between two Schwann cells is known as the neurolemmal node, neurofibril node, node of Ranvier. I go with node of Ranvier. Okay? So in a lecture, somebody was trying to pin me down, well, you know, you could put any of those for the lab exam answer. And usually if there's more than one name of something, I try to put both on a question on the lab exam. But if I don't do that, I'm most likely to use the node of Ranvier. And that's where the two Schwann cells come together and they don't completely, there's not a myelin sheath there. They partially cover the membrane, but in the formation of our action potentials, we need access to the extracellular ions. And the intracellular ions need to go out and the extracellular ions need to go in. So that happens at these nodes. So it provides the current formation occurs here, and then it jumps and forms here, and jumps and forms here, and it's another way of making that transmission much faster. All right. So that's why we have the nodes there. So again, on your diagram, I think the nodes are letter G. We said it 3 times today already. F is shown as the neurolemma, but would you also include the letter D because it's that outer cytoplasm as well, not just the plasma membrane. So D is supposed to be the Schwann cell or neurolemmacyte, but if you include F and D as the neurolemma. Any questions on that page? So D and F, both the underlying cytoplasm and the plasma membrane itself for the neurolemma. Okay, we're going to skip page 106. No, we're going to do page 106. It's the next 108 we're skipping. So we have these models of the neuron on the table and other ones and some on the chart. The cell body is this broad area here where we have the nucleus. And the branches, that would be letter F. The branches of the cell body are the dendrites. So they're not shown completely here. You'd have to look up in the painting to see the extension of the dendrites. These are truncated. One of my favorite words. I love words that sound like what they are. Like you can just visualize a tree that's been cut at the trunk. So these have been truncated, they've been cut off. But if they were to extend out, these are the dendrites, and their purpose is to extend the surface area for synapses to form. So those would be letter H on your diagram, the dendrites. Within the cell body, we have the nucleus, which is letter B. And it's very characteristic of cell body nuclei to have a very prominent nucleolus, sometimes described as an owl's eye.
Wait, D or B?
B would be the nucleus, and B is in boy, and C would be the nucleolus, where the RNA is. So the orange, whatever color it is, would be the nucleus, and the black circle would be the nucleolus. And then within the cytoplasm, we have, as I drew on the board, the typical protein synthesizing organelles. So we have Golgi apparatus, and we have the ribosomal ER. Some of them look like they have candy, little sprinkles glued to them to be the ribosomes. On our charts, these show as these dark clumps, kind of a teal in the back, center there, number 5. You can see those kind of bluish clumps. Those are the ribosomal ER or Nissl bodies. You can use either term for lab. A lecture question might be asking you, Nissl bodies are formed of which of the following organelles? All right, but for lab you could use either structure. And those look like E to me, the clumpy stuff. Neurofibrils are microtubules and filaments that are present in the cell. I don't really pay much attention to them, but that's letter G. Mitochondria, remember, are found in both the cell body and the axon. So mitochondria are providing energy, and they're really significant along the axon. It's a little bit of physiology, but remember I said we had to have some bare areas at the node for ions to move into the cell and out of the cell. Once that happens, those ions have to be returned to their original position to allow another action potential to be created. And it's usually against their gradient, going uphill, if you will. And that requires energy. So we have a lot of mitochondria in the axon to provide that. And another purpose is the neurotransmitters that are made here in the cell body have to be transported down to the axon terminal. And that's done along microtubules. And there's little arms that walk. I mean, they look like they're walking. They don't have feet, but they swing back and forth, all right, as they move down the microtubule, and that requires ATP as well. So we need mitochondria in the axon. The axon hillock is this tapered portion where the cell body is merging into the elongated process, which is the axon, and it has a lower threshold. Remember, we have voltage-gated channels that open when a certain number of ions flow by them. Well, there's a minimum number of ions that have to flow by. So I sometimes illustrate this by department doors or grocery store doors. Some of them have an electric eye. Some open because of the weight of the person walking across the carpet. All right, well, you wouldn't want a mouse or a rat running by causing the door to open. And so they may have a minimal weight, 30 pounds or so. And if you're 5 pounds running across, it's not going to open the door. All right. If you're 100 pounds, it opens the same way. So that's kind of the sensitivity of these voltage-gated channels to the current flowing by. So if it takes a current flowing by, say, of 50 millivolts along here, the axon hillock is more sensitive. It might only take 40. So it's going to open a little easier. But it would create the amount of current needed to go by the rest of the axon. All right. So on this model, it's the tapered region from cell body to the normal axon. So this would be the axon hillock right here. And that would be letter D. You can see the kind of the dotted lines there showing that segment. So D is in doorknob. And then we have the axon itself, which is letter M. Some axons have branches, you can see here, and a larger one on this larger diagram. These are called axon collaterals, and that's letter I. The axon axon ends by forming multiple branches, and these are the axon terminals. And the very end is dilated into a bulb or ball-like structure, and that's the synaptic bouton or end bulb. So that's what's enlarged at the bottom of the page, but the synaptic end bulb would be K, and the axon terminal J. Helodendria, again, is another name for that setup. All right, so we're going to do skip for now, page 108 and 109. We'll do that on Monday when we do our spinal nerves, because they come from the spinal cord. And let's finish up with 110, 111. This is what most of what I covered, or what I covered in lectures, most of what's on this page. In the past, I would have had it diagrammed on the board, but I did that in lecture already, so I'm going to look over at this chart. So this emphasizes that the spinal cord is not always a cross-section the way I do it. It's actually an elongated structure, and the gray matter is elongated and the white matter is elongated. We tend to look at it just as the cross-section. We also have spinal cord models. We'll have a few more out on Monday. Well, actually, we can bring them out now. This one is in situ, meaning in place. So these are cervical vertebrae and cervical spinal cord with the meninges wrapped around it and our dorsal and ventral rami exiting here. And here would be the dorsal root ganglion. Now you can also tell this is dorsal because spinous processes are dorsal. All right? If you just have that chart that's in front of you, so on your quiz on Monday, you will have one similar to this, if not identical. And you may get one like this with your name across here and the 6 questions down here, or it may be flipped and your name across here. So the first thing you're going to have to do is figure out which is anterior and which is posterior. It may not always present in the way I drew it. So there's 2 ways that you can define which is anterior and which is posterior. It's really easy if you have this chart because you have the dorsal root ganglion, which is obviously going to be on the dorsal aspect. Okay? The diagram in your lab manual is difficult to figure out, which is where the dorsal root is. If you have no dorsal root, what do you have to go by? The posterior gray horn, notice, extends all the way to the edge of the spinal cord. The anterior gray horn has white matter between it and the spinal cord. So look for that, because that's going to change everything. If you have it turned like this and you think this is anterior, then all your answers are going to be flipped. Okay? So you got to figure that out first when you're determining what's anterior and what's posterior. So when we look at this one, you can see how this gray matter goes to the edge of the spinal cord, whereas this does not. So this would be our posterior gray horn and the anterior gray horn. Okay? Now before we get to that, your lab manual also lists the 3 layers of the meninges. You've probably heard of meningitis, which is an infection or inflammation of those layers. There are 3. One is very closely attached to the spinal cord or to the brain. That's called the pia mater. And that would be letter C on your diagram. Mater meaning mother, for the protective role that these play. Then there's a gap filled with cerebral spinal fluid that we talked about this morning. And then we have the arachnoid matter. The arachnoid matter bridges that gap with the spidery filaments. Somebody thought it looked like spider web or like spider legs, I'm not sure which. So the arachnoid matter is the blue layer. The gap between the 2 is called the subarachnoid space. So the arachnoid matter is letter B, as in boy, and the subarachnoid space is in D, as in dog. I was thinking Derringer, but guns aren't politically correct anymore, so. And then the subarachnoid space is going to contain CSF, and then the arachnoid is directly and intimately adherent to the deep surface of the dura mater, which means tough mother. This is the outer one, and that would be letter A. along the way, in between where we have our spinal nerve components, remember the spinal nerves are going to be passing through the intervertebral foramina. So in between the intervertebral foramina, the pia mater is pulled out to the side and attached to the arachnoid and dura mater. And it creates a tent-like structure that's kind of sharp, reminded somebody again of those little shell teeth that chickens have. And so that's called the denticulate ligament. You can see it just starting here and then faintly through the blue on each side. You can also see it in this spinal cord model up front, okay, and a little bit in the spinal cord model here. So denticulate ligament is letter E on your diagram. Now it's not shown on your diagram, but the first phrase term is epidural space. And epi meaning around, remember our epineurium? So that means around. So between the vertebral foramen, the bone of the vertebral canal, and the dura mater, we have the epidural space. And that's typically filled with adipose tissue and blood vessels. Okay. Anybody had an epidural? Anesthetic? Okay. I have 3 kids with 2 are twins, so I gave birth twice. And I had an epidural both times. And that wasn't done in my cervical vertebra, okay? Because we have a spinal cord there. So remember in lecture this morning I said the spinal cord ends about the bodies of L1 and L2? So L3, L4 is typically where they go in, kind of on the side here, between the lamina, into the epidural space. And by that time the dural sac doesn't have a spinal cord. Sorry. It contains just the spinal nerves. And so if we look at this spinal cord here, you can see how the spinal cord ends here. And then we just have the descending spinal nerves going to each of their sacral foramina. Okay? This is still filled with cerebral spinal fluid. So an epidural would not go through the dura mater, but would just release the anesthesia into the adipose tissue around it. A spinal anesthesia or a spinal tap would go through the dura mater and put the anesthesia actually within the spinal fluid, or if you're drawing spinal fluid out to look for bacteria and an infection, it would take it from that location. Okay? So the epidural space all along the spinal cord is going to be between the bone and the dura mater, and it's not shown on your diagram. The word is there, but there's, sorry, my daughter calling. Savannah, can I call you back? So that's the upper portion under meninges and spaces. Central canal is right here in the center of the great commissure. Okay. So that would be letter N. The grooves that I didn't mention in lecture, well I mentioned but didn't name them, we have the anterior median fissure and the posterior median sulcus. So this is supposed to be, as a fissure, it's supposed to be deeper, but it's actually more shallow than the sulcus. The sulcus goes, the posterior sulcus goes all the way almost to the great commissure. So they named them the wrong thing. So I protest by not talking about it. All right, so now let's look at the gray and white matter. So back here we have the 2 posterior or dorsal white columns. You can use either term for me. And remember, they're only sensory. So all the tracts are ascending. There's no descending fibers. They're carrying information up to the spinal cord for balance. There's I don't know if you've heard of a guy, Oliver Sacks. He's written quite a few anecdotal books about his experiences as a neurologist. And in one of them, he tells the story of a woman who went into the hospital to have her uterus removed. And this was in the days when you would go in the night before instead of getting up at 4 o'clock in the morning. And she woke up and rang her panic button. And when the nurse came in, she said, "I've lost my leg." And the nurse was like, "It's right there." She said, "I know, but I can't feel it." And they just thought she was having hysteric attacks because she didn't want to lose her uterus. And so as the day progressed, she lost more and more, quote unquote, limbs. And they finally called in the neurologist, examined her, and she had no recognition. So if you close your eyes, My kids and I used to play this in the car. I wouldn't close my eyes on it when I was driving. But if it was summertime and I was wearing shorts, whoever was sitting next to me in the car would reach over and make a mark on my pen or my arm. And then I would, while I was driving, with my eyes open, try to do the same thing. Try to match as closely as possible, okay? So, I'll get one that's a really fat marker. And this requires the ability to figure out where your joints are doing. So make a mark, either on my anterior or posterior arm, okay? So now I felt that, and I have to keep that in memory. I have to realize where my arm is and what this arm is doing. My eyes are shut. And come back to that memory that I had and I was off by about, but I waited a while. It's also that ability that allows you to close your eyes and, do this or touch your nose, all those things they test you for, I assume, when you're pulled over for drunk driving. And that joint sense is what she had lost. And they finally discovered that she had had a viral infection that totally destroyed her posterior white column. She never got it back. And so at first she couldn't even walk. To sit up, she had to look at herself in the mirror. And even now, when she walks, it looks like she's drunk because she doesn't have that information coming back to her body, to her brain. She said the only best time that she feels alive is when she's in a convertible and she feels the wind blowing against her body parts, her head or hands or whatever, and has an idea of where they are. So that's what is carried on in the posterior white column. Right? That would be letter F. Lateral white column is G. And this is mixed. We have distinctive motor and distinctive sensory tracts, information going down or up. And then anterior is also distinctive motor and sensory, and that's letter H. Okay, then we talked about the gray matter already, so here's the anterior gray horn, made of multipolar cell bodies. That would be L. The lateral gray horn, right here again, primarily seen in the thoracic region, is K. Posterior gray horn is sensory, that's J. So the sensory neurons coming in here, many of them will synapse with the cell bodies of additional sensory neurons located there. M is the gray commissure, in front of and behind the central canal. And then we have the dorsal root or posterior root here. That is only sensory because it's going into the posterior aspect. And that will be O. The anterior root is P. And the dorsal root ganglion is R. Now it's hard to see that as a dorsal root ganglion because it's still wrapped up with dura mater, and it's kind of difficult to see where it's actually separated. Okay? Now on this diagram is absent the rami, but I want you to know the rami. So here would be the dorsal ramus going to the middle of the back. Remember how small that is? And then all the other neurons going to your upper extremities and the trunk are in the ventral rami. So add dorsal rami and ventral rami. You just add it to your drawing if you want to. And this is part of the sympathetic nervous system, so we're not doing any part of that. We'll have to worry about that part of the chart on here. So again, your quiz on Monday will be that diagram with 6 structures marked on it, asking you to identify them, and let me know what the sensory, motor, or both. Okay?
Dr.
And then we'll
Lindsay,
pick up with final nerves on Monday. Yes?
can you repeat what the longitudinal grooves were again?
Sure. This would be the anterior, median,
That would be S,
the anterior median fissure would be T,
T,
and posterior would be S. Alrighty. I'll tell my daughter and see if she wrecked the car. She probably just won't.